to the 17th Annual Genetics Prize, honoring a leading scientist or group of scientists for groundbreaking contributions to any realm of genetics research. We're delighted to be here at Baylor to present the prize. We also would like to add our condolences to those of many of you here on the recent passing of former President Dr. Butler. I'd like to acknowledge also the founding vision of my late husband, Peter Gruber, in establishing these prizes, as well as his passing in 2014. After leading a long and productive life, many know his story. He was born in Budapest, and after fleeing Hungary in World War II, his family settled in India, where he was educated there by Jesuits in the Himalayas. Later, he came to the US and he went to work on Wall Street. It was his success as an asset manager that allowed us to establish the Gruber Foundation and to create the prize program a few years later. To continue this legacy, we reached an agreement with Yale University in 2011, establishing the foundation there where the science prizes and the human rights programs continue. Yale hired our very own Sarah Rea as executive director to guide the smooth transition of all the foundation activities there, and I will remain a lifetime trustee. Today we're here to honor Stephen Elledge for his pioneering work in the discovery and characterization of the molecular mechanisms of the DNA damage response pathway. Please let me tell you first a little bit about the company he is keeping. The Foundation's prize program was established in 2000, and it now presents 3 annual $500,000 prizes in the fields of cosmology, genetics, and neuroscience. Each prize recognizes achievements and discoveries that produce fundamental shifts in human knowledge and culture. On October 13, in a ceremony at the 4th Annual Cosmology Conference at Yale, the Cosmology Prize will be presented to Sandra Faber. The Neuroscience Prize will be presented at the annual meeting of the Society for Neuroscience in Washington to Joshua Saines. Nominations to all 3 2018 Gruber Prizes are open until December 15th of this year. We'd like to also proudly mention that the Nobel Prize Committee has announced in the last 2 days confirming prior Gruber Committee selections, former Gruber recipients in areas that have been announced so far this last year. So we have to say what the financial world says, that past performance is no guarantee of future results, but we have our fingers crossed. Returning to genetics, we are proud of our illustrious laureate roster, and we're very pleased to be adding to it today. The 2017 prize recipient was chosen by our distinguished advisory board. We deeply appreciate the knowledge, commitment, and enthusiasm that these advisors bring to the judging process. We would like to also thank the Genetics Society of America for their long and ongoing support of both the Genetics Prize and the Rosalind Franklin Young Investigator Award, which is presented to honor the groundbreaking contributions of Dr. Rosalind Franklin and to inspire and support new generations of women in advanced genetics. I'd like to now bring back Huda Zogby, who will read the official citation for the 2017 Genetics Prize and present the scientific accomplishments of today's recipient. Professor Zogby.
Citation that Steve will explain. I will read it from my comments here. The Gruber Foundation proudly presents the 2017 Genetics Prize to Stephen Elledge for discovering and characterizing the molecular mechanisms of the DNA damage response pathway in eukaryotic cells, findings critical for understanding pathogenesis and developing therapies for cancer and other diseases. Elledge used clever genetic screens in yeast to identify mechanisms by which cells sense DNA damage and direct effector molecules to initiate efforts to repair the damage and halt progression of the cell cycle until repair is completed. He also showed that this process is conserved in mammals with orthologous genes playing similar roles. Mutations in many of these genes drive cancer in humans, while others contribute to premature aging or neurological disorders. So to tell you a little bit of background to put Steve's work in context, what happens if there is an emergency? Let's say a neighbor collapses in their yard and you can't revive them. What do? You'd probably call 911, and an ambulance would come within a few minutes to take your friend to the hospitals. It seems pretty straightforward, but for this system to work, there are a lot of components that have to be in place. First, you need to have access to a phone. There have to be phone lines to transmit the call. There has to be an operator to direct the ambulance, and there have to be roads to drive on, and an emergency room to drive to. If there is a breach at any point, say if the roads are flooded, or the driver gets lost, or the hospital loses power, we're in big trouble. The situation is analogous to what is happening in living cells. Our DNA is actually fairly easily damaged, whether by exogenous forces such as ionizing radiation, or by endogenous processes such as DNA replication. Normal cellular metabolism causes oxidative stress, resulting in thousands of DNA breaks in each cell of our bodies every day. The reason we're able to remain healthy is that the body is constantly repairing our DNA through the DNA damage response system. In the 1960s, Evelyn Whitkin at Rutgers University has discovered that bacteria are able to sense when the double helix is broken because of a protein called REC-A. It scours the cell for the single-strand ends that result from DNA cleavage. REC-A forms a filament around the single strand and initiates what's known as the SOS response. 20 years later, when Steve did his postdoctoral training in Ron Davis's lab at Stanford, he set out to identify the least ortholog to REC-A. He didn't find it, but he did discover UV-responsive elements that allowed him to undertake genetic screens for factors that were activated by DNA damage. When he came to Baylor as a young faculty member, he set out to discover how the system worked in mammalian cells. He hypothesized that the damage response had to be initiated by some sort of damage signal, which then would have to be amplified and then translated into repair activities by effector molecules, much like your emergency call translates into activity by systemic emergency response team. He turned out to be correct, and he spent the next 20 years elucidating how over 1,000 components are involved in these three stages of the response. Steve discovered that when cells find damaged DNA, they activate checkpoint proteins that halt cell division until the repair is complete. He also discovered that the cell responds differently to DNA replication stress and DNA damage, but that the two response pathways share many components. Many of these damage response factors, such as BRCA1, have become notorious for their roles in cancer. In fact, Steve has recently embarked on studies of how chromosomal abnormalities drive cancer, tackling questions that have been unresolved in the field for over a century. Steve is a prolific and generous scientist, an outspoken advocate for scientific research and education, and a dear colleague who has influenced my own research in the past several years. Steve's use of genetic screening in cancer, summed together with his postdoc Trey Westbrook, who is now faculty at Baylor, inspired my lab to use the same strategy to find proteins involved in neurodegeneration. There is no doubt that Steve has opened many fields, ranging from DNA damage to cancer and immune responses, and inspired many others. We are delighted to welcome him back to Baylor College of Medicine to celebrate his Gruber genetic class.
Let me have you in the shot when I shake hands. Yeah, and everybody right here.
Thank you. Great. I'll just set this up here. Great. Well, thank you. Thank you very much for this award. You know, I've won a few awards in the past, but this was really extremely special to me because I'm a geneticist, and this is really the ultimate genetics prize. And I consider myself a geneticist through and through, and I think about genetics. It's not a tool; it's a way of thought, and it's opened up so many avenues for me in science, and that is in part why I work on so many different things. So this is particularly special to me, and I'm really happy to receive this award. Also, I'd like to point out that my postdoctoral advisor, Ron Davis, won this award in 2011, and my next door neighbor from Baylor, when I first started here, Dave Alice, won it I think a year ago or 2 years ago. So I have known and admired all well, Dave's not a geneticist, but it's I've known and admired many of the geneticists who won this award, and that also makes it very special for me to follow in their footsteps, because many of them are really great geneticists. And finally, I'd just like to say, before I get started, that the fact that this award is being held here at Baylor is especially great for me. I mean, you know, usually this is at the Human Genetics Society meeting somewhere in Philadelphia or DC, but this year they decided to have it at Baylor, and it's just so great to be back here where it all began, and the screens that we did that first elucidated the DNA damage response really were done by Jing Zhou, my first graduate student, who joined my lab and was just a tour de force. She was a force of nature, really, that came in like a whirlwind and developed these genetic screening methods. I don't know, Jing, you must be here somewhere. I don't know where you are, but thanks. She came in and really set that stuff there, and really got it going. And that really was the beginning. And they were beautiful genetic screens. And she published 2 papers on them, one in Genetics, the journal Genetics, and another in Cell. And it really set in motion everything that I got this award for. So it all started here in Baylor in the biochemistry department. And so coming back here to receive this award at Baylor just makes it even better. So I couldn't have imagined a better way to receive this. So let me just switch the projector here. So what I'm going to talk about today, I'm not going to talk about the DNA damage response because I've spoken here relatively recently about that. So I'm going to talk about what I'm calling microgenetics, and I just made that word up. So it doesn't really mean anything, but what I'm going to talk about is what we can learn from bits of genes. And so this is, I used to teach genetics here at Baylor, and I found my old genetics slides, and the first slide I started with in genetics, my lectures with, said that genetics is a study of how proteins interact, fold, and function. I would amend that to include RNA somewhere off to the side nowadays, but that's the way I always thought about genetics. And it's really about the study of proteins and what happens when you interfere with them and what phenotypes that you get. And of course, you know, there are all kinds of proteins that come in all kinds of shapes and sizes, and they build large complexes. And if you look within a family of any particular protein, these are PDZ proteins, you can see that they have lots and lots of different domains and regulatory motifs. And so one of the key questions is how can we begin to sort out the roles of these domains and regulatory motifs that these proteins possess. And so I'm going to describe a set of tools that we developed and how we've used those tools to ask questions about three different parts of biology. One is about protein stability, and another is about transcriptional regulation, and the third is about the immune system. So just to start out with protein stability, so this is very basic science stuff, and the question is about proteostasis. That is, how do you determine the abundance of the protein in the cell? And it's a struggle between protein synthesis and protein degradation. And in particular, we were interested in protein degradation based on some work that I did here in 1996 that had to do with the ubiquitin proteasome system. So that's the major system that controls protein stability. And it works in a sort of classic cascade of three proteins. E1 is the ubiquitin activating enzyme. And in an ATP-dependent process, it activates a small protein ubiquitin to form a bond with a cysteine, sulfhydryl group here. That's then transferred to E2 ubiquitin conjugating enzyme. So there's one E1, and there's about 40 of these E2s. But the key is how you decide which protein to put this ubiquitin on, because the ubiquitin signal is a signal for degradation. And that's accomplished by the E3s. These are the adapters that work together with the E2s to find the right proteins to ubiquitinate and destroy them. And so the key are the E3s. So let me give you some background about the E3s. Shown here. So there are two general classes of E3s. There's the HEC domain proteins, of which there are only a handful. And these are some of the first E3s to have been identified here. And then there's a much larger group of these ring domain proteins. And these were, many of these were identified here at Baylor. In particular, they fall into two general categories. One are the colon ring ligases, and the other are basically monomeric rings. And so when I was at the biochemistry department, we discovered the first of these colon ring ligase complexes, which are FOX proteins in the SCIP1 gene that we found genetically studying in yeast. And we realized that these were adapters that sort of plugged in to this machine like a socket wrench and would change the specificity of this machine towards different substrates. And we also found evidence for the CRL2 and CRL5 protein components of this ubiquitin ligase family. And then together with Wade Harper, we figured out the assembly of the entire complex and that they acted as ubiquitin ligases. So these proteins for CRL1, colons are these scaffolds here that assemble the E2 and the adapter proteins. These are FOX proteins, and as I said, work from with Wade Harper largely, and several other people helped elucidate these. Now, one of the other advantages of this system is that there is an inhibitor that will inhibit all of these ligases by blocking a modification on the ligases. So this is a drug that we use in this to turn off the colon ligases, and that's very useful. So, as I said, the FOXes proteins, they come in a lot of different flavors, about 80 of them, and each one of these is a landing pad for different substrates. So they're basically integrating into the proteome and trying to model it. So you can think about E3 ubiquitin ligases in much the same way that you think about microRNAs, because microRNAs filter the transcriptome. And in the same way, E3 ubiquitin ligases filter the proteome. So there are about 600 ubiquitin ligases, and the question is, how do you hook them up? How do you find out which E3 recognizes which substrate? How do you link them up? And so this is a large fundamental problem because it's really easy with microRNAs. because you can use the sequence of microRNA to identify the target for the messenger RNA. But E3s are a completely different problem. So how do these E3 ubiquitin ligases work? Well, they recognize something called a degron. And this is a localized element that's on a protein that's recognized by the E3. And they're typically short linear motifs. Sometimes they're modified by phosphorylation or acetylation or other modifications. And they're transferable. So if you take a degron and fuse it to a stable protein, you'll confer instability on that protein. So now we were interested in this problem as soon as we discovered the colon ring ligases, which is probably the largest family of ligases. And a lot of people have worked on these and tried to identify various substrates. But you can see there's really only a handful of these. There's about, I think, about 13 general sequence motifs that have been found that correspond to different ligases. And that's the sum total of what we know in the 20 years since we discovered this. So in order to get at this, we developed a genetic method to assay the stability of a protein. And so that we could look at lots of proteins at one time. And so basically the idea is to fuse your protein or degron to GFP. And so you can look at it confers its stability on GFP, and so you can follow it. But the problem is that you can't really do this if you say put it on a virus and infect a cell to make a reporter cell line, because wherever the virus lands, the amount of transcription is different. So if you just put GFP on a lentivirus and put it into a cell and infect it, you'll get 2 orders of magnitude difference in the level of expression based on where it lands. So transcription completely interferes with this. And so in order to get around this, we developed this system that has an internal reporter. So this is a DS-red, which is a red fluorescent protein. And then we express the GFP from an internal ribosome entry site, or an iris. And so it turns out that since they're made on the same transcript, it doesn't matter how much transcript is made, the ratio of GFP to RFP is constant. And so that is a readout for the overall stability of the protein. Okay, so the ratio of this fusion protein to DS-red normalizes for this. And these are some papers that we published, the first 2 in 2008, and then another paper in 2011 on this system, using it to fuse to all human proteins to do experiments to look at the stability. Now, you can look at full-length proteins using an ORF library, but you can also use short peptides. And so that's what I'm going to tell you about today, is how we approach this problem of sorting out these domains and regulatory motifs. And that was by generating a synthetic representation of the human proteome in short peptides. And this takes advantage of the ability to program oligonucleotide microarrays to encode anything you want. So we made a tiling of the entire human proteome in about 90 amino acid long segments that are tiled every 45 amino acids. Okay, so you can make this oligonucleotide representation. You can then PCR out and clone it into this lentiviral vector using these peptides to GFP. Then you can infect them into cells and do fact sorting based on the RFP-GFP ratio. And so the things in the top bin here, bin 4, are very stable, and the unstable ones are here. So this is what we did. And you can just PCR out the inserts and sequence them to figure out what peptides cause instability. And that's shown on this slide. So this is the unsorted library, the GFP to RFP ratio. And then we can sort them in these bins. And so this is the unstable bin, this is the more stable bin, the most stable bin, and this is what it looks like relative to the starting GFP. So this is what the GFP RFP ratio looks like with GFP, and this is the unstable. So you can see that we really clearly distinguish these two bins. Now, I also want to point out, I won't talk about this, but if you look at where this goes, it's a little, the center of this is a little left, the 10 to the third, but over here, we have a lot of things over to the right. It turns out we can find peptides that actually stabilize this. That's something we're following up on, but I won't talk about it today. Stable bonds. But I just want to point out that of this group of unstable proteins, there are two general pathways to degrade proteins. One is the lysosomes, which is autophagy, and the other is the proteasome, which I alluded to earlier. And there are inhibitors for both of these. And so if you add an inhibitor to autophagy, that's in blue here, you can see that the blue line shifts a little bit to the right of the control line here. So there's a small number of things going to autophagy. But the vast majority of these things are stabilized by inhibitors in the proteasome. So the vast majority are going to the ubiquitin proteasome system. Although sometimes things go through independently of ubiquitin. We have a way of looking at that. So we were particularly interested in, at this point, in looking at colon ring ligases. And as I said, there was a drug that can inhibit this group in particular. And there are a couple of atypical colon ring ligases, and then one very unusual one, which is the anaphase promoting complex, which we won't talk about today, that's dedicated to mitosis at some level. So we wanted to find the substrates for colon ring ligases as our first foray into understanding this. So the way we did that was that we took the inhibitor of colon ring ligases, MLN4924, and added it. And what you can see is that you see a significant shift. It's about 8% of the peptides go through there, and you can just sort these out. And you can toggle it back and forth, kind of we call ping pong sorting, to get a pure population of colon ring ligase substrates that now the whole pool, when you add it, shifts over to the right. So these contain a variety of substrates for different colon ring ligases. So how do we figure out how that works? Well, now that we have just a pure pool, we can then interfere with each of the colons, which are the scaffolds, and to see which peptides now respond to turning off just one of them. And we do that by transfecting a dominant negative colon substrate, or sorry, a dominant negative protein fragment that interferes with the assembly of the complex. So you can see here that not much happens with col1, but col2 and col5, which are related proteins, you can see a significant number of these peptides are stabilized here. And then for col3 and col4s, of which there are two, you can see they each do a little bit, but nothing at this level. So we decided to first follow the CRL2 responsive peptides. So we purified these out here. We sorted these to purify them, and then we took 17 of these clones that respond to a dominant negative col2. The dominant negative is just the first half of this, so it soaks up the adapters, but it doesn't bring in the E2, and that's how it stabilizes. And so we took 17 clones, and then we made a CRISPR library to some of the colon ring adapters, and this sorted for individually 17 mini screens to find out which adapters would stabilize a particular peptide fused to GFP. And that's shown here. And of these 17, you can see they fell into groups. In particular, these are each different adapters. Probably these two form a complex. But we're going to focus on this one, the KLH, Kelch domain DC3 family. And when we looked at which peptides were stabilized, it turned out that all of the ones that were stabilized by interrupting this Kelch domain protein all ended in a glycine. And some were very short peptides. So we were thinking that perhaps these ligases are recognizing the ends of proteins. And so in order to test that, we mutated them. We took a glycine to alanine mutant. And what you can see, and also glycine to aspartic acid, and what you can see is that mutation of these stabilizes this particular protein, although the G to A mutant only partially does it. We also just added a C-terminal extension, a few extra amino acids on the end, and that also stabilizes it. So we really do need the end of this. And we found another ligase here that recognizes 2 Gs. It's a related protein, but I won't talk about that today. But also requires the end. So this made us think about the end of proteins and where would be the best place to look for degrons. And we thought, well, maybe there's a specific pathway that likes to look at the ends of proteins. And when you think about protein structure, if you look at the percentage of disordered protein regions, they're greatly enriched at the ends of proteins. So the N-terminal and C-terminal 40 amino acids here have a high degree of disorder compared to the internal regions. And it's also true that the N and C termini of proteins show less conservation across phylogeny. So that suggests that they're more free to evolve, and they're more or less constrained to evolve regulatory motifs than if you're embedded in the structural part of the protein. And that makes a lot of sense if you just think about how histones are regulated, right? Because all the regulation is on the arms. So, so we thought, well, maybe the ends of proteins are going to be great sources of finding these regulatory motifs. So now we're not the first people to think about this. There's a whole area of research pioneered by Alex Varshovsky, who developed this concept called the end rule, he called it. And his data was generated in yeast, and he showed that different N-terminal amino acids confer different half-lives on proteins. Most N-terminal amino acids start out with methionine. Sometimes they're cleaved off, sometimes they're acetylated. But in his system that he worked this out in, he was actually cleaving off the N-terminus by fusing ubiquitin to it, which can be cleaved off. And so he could start a protein at any amino acid. So this maybe is more applicable to proteins that undergo proteolysis. But there is a whole area there on the N-terminus, and he discovered these E3 ligases called UPR proteins that recognize particular amino acids that he calls the N rule. So there's a family of 5 of these. And so we were wondering whether or not there might be something similar for the C-terminus of proteins. And so in order to ask that question, our original library had everything in it. It had the entire proteome. But we made a different library that focused just on the C-terminus. So we made the C-terminal 23 amino acids of every protein in the database, human protein. And we cloned it into this vector that I described earlier, and we started doing analyses on this. We sorted it into bins, as before, and then sequenced them. And so what we found, this is sort of general observations, are that in the most unstable bin, bin 1, versus the stable bin, bin 4, you saw an enrichment of certain amino acids. So anything that's hydrophobic, especially the bulky hydrophobics, were enriched in this. And just the opposite was true in the stable proteins. They were highly enriched for acidic residues, D and E, and they avoided hard hydrophobics. So this is plotted out here. So it turns out that the number of hydrophobics in this 23 amino acid region help determine a lot of the stability. So the greater the number of bulky hydrophobics, the less stable you are. This is an index we call the Protein Stability Index, or PSI, and basically it tells you, it's a weighting factor that tells you approximately what bin you sort into. So low numbers are the most unstable bin, higher ones are more stable, and the more acidic you are, the more stable you are. And that just comes out of this. Now we don't know why that is, but it's just a general rule, and that's something we need to understand eventually. But let me move on to how we did the computational analysis to determine potential C-terminal degrons. And the first thing that we do is we identify motifs at the C-terminus. So, and this is just a GG motif, which is unstable. And what we do with this is to look at the PSI score of all these diamino acid motifs when present at the C-terminus as opposed to internally. And so we can go back into our data sets and we can pull out all the peptides that have a GG either at the end or in the middle, and we can say, well, what's the average stability of that? And then we can just subtract them to give a score. Okay, so we do that to calculate this score, and then we repeat it for every possible combination of two amino acids, and we plot them in a graph. So here's just another example of a motif, RXXG, and this is what it looks like when it's at the end. This is what it looks like when it's in the middle of a protein, and you can plot these things out. So we're looking at this differential stability and looking for the things that are down here that show differential stability when they're at the end. And so what you can see here is that when you expand this, is that there are a number of different motifs, and here we've just color-coded them. These are GN motifs. This is RXXG in green. There's also EE motif that's unstable here, and various other motifs. And this just shows you what they are, where they're located, and we have a variety of these different motifs that are showing up. Now, we don't know that they're all being recognized by different E3 ubiquitin ligases, but some of them clearly are. So, and there's our EE, we'll come back to that in a moment. And so this just shows you what the differential looks like between bin 1 and bin 4, the stable bin, and the unstable bins for a variety of these in these box plots. So you can see that the average stability really does change. So now further examining this whole pile, this whole group of ubiquitin ligases at the end, for which ones are regulated by colon ring ligases. So we went back to our trick with the colon inhibitor, and we sorted for the ones that were unstable. And when you look at the amino acid distribution of these peptides, for the ones that respond to colon ring ligase inhibitors, you see that there are certain amino acids that are highly enriched in the regulated, colon-regulated population. And this is what they look like here. And so each one of these is potentially regulated by a colon ring ligase. And you can see that we get our GG here and RG, which are recognized by two different adapters. So we can then go back and do our trick to figure out which of these colon complexes target these C-terminal degrons by using these dominant negatives here, where you just express the first half. And so we can take the entire colon-responsive group of C-terminal peptides and figure out which ones are actually regulated by which colon ring ligases. And so you can see here, now we can see col2 again is showing some effect, but also now we get a strong effect by col4. And if you look at this, so this is what I showed you earlier for the colon ring ligases. You can see that now, in addition to G and a strong R at -3, we're getting this acidic EE motif here. And when you look at col2, when you interfere with this population with the dominant negative col2, it picks out the G. It also picks out a histidine here in the middle, but now you're losing the R at -3. You gain a few other motifs that now come out more strongly. When you do col4 dominant negatives, you can see that now you pull out this EE motif and this R at -3. So col2 and col4 complexes are targeting the C-terminal motif. So we want to know if these actually are at the C-terminus. I mean, we predict them, and we're trying to figure out what they regulate. So here we took a few examples of these motifs, and we can show that this EE and the RXH and RXQ motifs are regulated by dominant negative col4. So you can see that you shift from the control to the right to the more stable versions. And then to ask whether or not this is actually important that this motif is at the end, we added a C-terminal extension of a number of amino acids. And what you can see is that now they're all stabilized, and whether or not you have the colon inhibitor present. We then did an alanine scanning on these, and we mutated the acidic residues to alanine, or this R motif here. And something funny happened, which is that they're still unstable, but they no longer react to the colon inhibitor, but they still react to the proteasome inhibitor. So by chance, we created a new degron here. And so this is sort of a cautionary note for doing alanine scanning mutagenesis, because it turns out that there's actually an alanine degron, a C-terminal degron that you can mutate to. So, just to go forward with this, we focused mostly on the culinary ligases, looking at the C-termini of proteins, and we wanted to know whether or not there were other family members, other E3 ubiquitin ligases besides these. So we were interested in what else here might be regulating these proteins. So we can subtract out all the colon ring ligase substrates and look at what's left. And when you do that, you see that there's still other motifs that are not regulated by colon ring ligases. And these are shown here, including this AA motif, A at the end, or A at -2, and valine at -2, and a variety of others. So this is sort of a computational approach to identify regulators of the C-termini of proteins. That took us to now just sort of look in general at how the ends of proteins are regulated by the C-terminal amino acid. So in this particular analysis, we just took the 4 bins of stability, or 5 in this case, and looked at the frequency of each amino acid here. And what you can see here is that if you have a glycine at the C-terminus, that it's enriched in the unstable bin, and it's depleted out of the stable bins. But the rest of the amino acids sort of bounce around the origin there. So glycines in particular are unusual. And so we looked across all proteins in the human proteome to look at the distribution of amino acids at different positions relative to the end. And what you can see are that for most positions, they're sort of hovering about the average, which is 1. But at the C-terminus, there's a very large dispersion an increased variance in what amino acid is at the C-terminus. And in particular, glycine, again, is the most depleted residue. So it's only present at 60% of the frequency that you see it at all of the positions in the C-terminal peptide and across the entire protein as well. So we think that this is an indication that during evolution, proteins have been avoiding creating these degrons at the end. And so, but that's just a hypothesis. It's hard to actually prove that over evolution this is happening. But it turns out it's not just in humans, it's in all eukaryotes. So you can see here that now looking at all eukaryotes, there is a depletion of G, and this does not happen in archaea or bacteriophage, but it does happen in human viruses. So proteins across all eukaryotes are avoiding having glycines at the end. And, but we don't really know if it's due to the fact that there's a degron there or there's some other aspect of biology that the C-terminal glycine is participating in that is driving this. However, there is a prediction, and that is that if it's really the degron that's driving this depletion across the C-termini of all proteins, then that's likely to be that the remaining glycine-ended proteins will be depleted further for the actual amino acids that are required for the degron. And so in order to figure out whether or not that's true, we had to figure out exactly what the degron needs. And so when typically you go about this by doing alanine scanning, although I did just mention that that's problematic in this case. But if you take a particular unstable peptide that's degraded by this glycine here and do alanine scanning, you see the glycine has some effect looking at this heat map here. And then if you then mutate the C-terminal glycine to every other amino acid, you can see that when it's glycine, it's fine, but everything else really interferes with it. But in order to know what the real degron is, you have to do this to the whole degron. So then we mutated every amino acid, or every residue to every other amino acid, and then you can really figure out what was allowable and what wasn't. Okay, so you can now take this information and separate it into two groups. At the -2 position, what amino acids are allowed and which ones are disallowed. And you can take those and now take them as a group and ask of the remaining glycines at the end of proteins that exist in proteins, are they random? Does it matter whether there's a degron there or not? Which you predict would be true if the glycine was avoided for a reason other than its role in degradation. Or is there a selection against it? And so these in red are the residues that are permissive for the C-terminal degron, and these are the ones that are not allowed. And what you can see across all these eukaryotic species is that there's an enrichment for the ones that are disallowed, and the ones that make a degron are even further depleted. So we think that this means that the actual presence of a degron is shaping the distribution of glycines at the ends of proteins. It has some role in it. I can't say that it's 100%, but it's definitely affecting the evolution of the end. So just to summarize this part of the talk, we've identified something that we think might be analogous to an end-end rule that operates at the C-terminus. There are multiple different colon ring ligases that recognize different motifs that are required at the C-terminus. And then there are also a number of, and many of these we haven't identified the actual adapter for, but there are also a group of non-culs. And so I just want to say that this is a work in progress, that there are a lot of things that we can do with this. and just point out that you know, the limitations of this. One is that this is all done with cytoplasmic proteins. We now can redo this analysis by forcing the proteins substrates into the nucleus to see if there's a different set of ligases that work there. And we can then further carry out the same sort of high-resolution analysis now on the N-terminus, which has not really been done, to figure out if there really are other factors other than the N-terminus that controls the stability of proteins at the end. And we think we can use this to identify hundreds of Dagron sequences, and this is going to inform the biology of protein stability, because when you find a protein, you can now just look up whether it has a Dagron or not, and try to figure out how that might be regulated. And I think we need to generate this sort of a map. So. And so this is just one thing that we can do. I have a couple other examples of what we can do with these bits of genes, and I'll just go through them briefly here at the end. And so one other application of this peptidome technology is to look at transcriptional activation and repression. This is pretty straightforward. We can make a library where we fuse these peptides to the Gal4 DNA binding domain, put it under inducible promoter, and make this in cells. We can then ask for how it activates or represses reporter gene constructs. So down here we have a reporter that's largely off, and we're turning it on. So we sort for peptides that increase the expression. We can do the same thing for repressors. Taking a strong promoter, CMV, and turning that off, and sort for things that are now at the bottom part. And just to give you a flavor for what we see with this, here are some zinc finger proteins that are known activators, and we get 3 or 4 peptides scoring in this region that's been thought to be the activation domain of these proteins. We're identifying things that we know are correct. Here's 2 other positive regulators, P53, and P73, which have known activation domains at their amino terminus that we pulled out of this screen. And we also found a large number of repressor domains, and many of these have been identified previously as crab domains. This is a well-known domain, but our PFAM search actually identified about 330 domains, most of which are crab domains, but there are 80 different types. And so there are a lot of novel repressor domains that we've identified here in this sort of systems analysis of these domains. And this just shows a number that have been highly characterized previously, HP1 and the crab domains, and the chromo domain and the chromo shadow domain, which is shown here. So there's a lot of new domains. I'll just point out, we found one particular peptide that was interesting in a protein of unknown function called KIAA1226, and it has a repressive peptide that takes the active reporter and completely turns it off, and adjacent to that, just 2 peptides away, is an activator. So they're set up next to each other, and this one in particular was interesting. So if you look at this, there's a little blip over here. So everything's really shut off, except there's a little blip of high activity. And so we said, oh, well, there's a problem here. We sorted it away, and it came back. And then we sorted for this, and then it all shifted over to the left. So we thought, well, there must be something that's changing here, and maybe it's the cell cycle. And it turns out that this repressor, we think, is turned off during mitosis. And to look at that, we just took unsynchronized cells in red and then added a microtubule poison called the cortisol. And you can see that it shifts way over to the right. And if you release it, it shifts even further, and then it goes back down. So we think that there's probably a regulatory circuit that is turning on genes on metaphase chromosomes, because that's all that's there at that time. You think of metaphase chromosomes as being turned off, but we think there's actually some expression, so we're following this up. We've done a genetic screen. We've connected this to some proteins in the TRIM family and the SUMO, and so we're trying to work out whether or not this is really working at the level of activating gene expression in mitosis. So that's where that is going. And then the last example I want to talk about is using these peptide fragments to interrogate the immune system. And when I first started thinking about this process, I was looking for autoantibody responses that might predict cancer, and there really wasn't a good way to systematically go after it. But people had previously done phage display of ORFs fused to different bacteriophage coat proteins to search for things. But it was a very laborious process where you had to do panning, and it wasn't genome-wide. But we realized that we could take these fragments of proteins that do cover the whole proteome and clone them into phage display and make a library where you could take blood or antibodies from a patient's sera, immunoprecipitate, and figure out by sequencing which fragments are being recognized by the antibodies. So we started this out by looking at human proteins, but we didn't realize that we could look at pathogens. So then we made a library that covered all human viruses. And the idea here was to, and these are shorter, these are about 56 amino acid fragments, but they're tiled across all these. And the idea here is that maybe we could look at the history of viral infection in these cells. And this worked out really well. We developed this tool called VirScan, and I won't take you through everything we've done with it just because of time, but this is just an example of serum from a number of different adults and children, and each one of these columns is a different sample, and each one of these rows is a different virus. And you can see that each person is different, and that, for example, the top row is Epstein-Barr virus. Almost all adults are infected with Epstein-Barr virus, but very few children have. But apparently they're going to get it. This is known as a kissing disease, so, you know, these kids are all 10 or younger. So, basically, we can identify these viral exposures or specific immune responses that are associated with disease. And for example, if you think a virus is leading to a particular syndrome, you can then take patients with and without the syndrome and say, what viruses have they been exposed to? And there are a lot of uses for this. And one of them is to really look more deeply into the immune system. So what we have the ability to do is to look at the actual epitopes that different people are recognizing who've experienced the virus. And this is a group of people from all over the world. And this is just one protein to adenovirus, C virus. And this is each peptide tiled into the whole protein. What you can see is that these two peptides are recognized almost by every individual. And that's true for a number of epitopes. We call these public epitopes. It means that your immune system is actually behaving the same way towards these proteins. We're very reproducible. And the question is, well, okay, it's the same peptide, but is it really binding the same, using the same amino acids? And so you can do alanine scanning with a single, double, and triple. And what you can see is that it turns out that they all are recognizing the same amino acids largely. And so, that begs the question of whether or not they're making very similar antibodies or not. And so we're now trying to find the antibodies from different patients that recognize these different epitopes to see, because if we're all making the same antibodies, even though this is a very complex way of assembling an antibody, and there's a lot of mutation and rearrangements that go on. If they end up with the same solution, that's going to be quite surprising. So we're interested in that. You can also just follow kids. This is a group of kids that we were following because they're prone to diabetes, but in the meantime, we found a lot of things about them. So when they're born, they have all these antibodies from their mother, and then by the first year, they go away, and then you can see that they are sporadically, this is 48 kids, but they're showing up with different infections to different viruses at different times. So you can just follow their exposure throughout their childhood. One of the more interesting things was that we discovered, we were actually trying to get an assay to look at, some viruses are easier to detect than others. We had trouble detecting measles, probably because most people are vaccinated when they're very young, and then they're never challenged again. So the titers of their antibodies go way down. They still have an immune memory, but not much of one. But we were able to get a group of kids from a Bible Belt in the Netherlands. So they have a Bible Belt, believe it or not, in the Netherlands, and so their kids don't get vaccinated, because their parents feel that that God should decide who gets sick or not. But after that, they're fine to let us have their serum to analyze. So we were able to get these kids who had measles, because there are these many epidemics that just sweep through the school, right? So we got serum from them, and we could very clearly see measles virus. But then we looked at all the other viruses, and it turns out that in the strong cases of measles, the titers that we had before and after, the titers of, this is a number of peptides identified and a heat map, so blue means they go away, that the antibodies to a whole bunch of other viral proteins drop in these kids. And it turns out, this had actually been seen for a couple of viruses that people had looked at, but they had no idea that it was completely general. So it turns out that the measles virus wipes out your existing memory. And so kids that get measles after that, they're much more susceptible to other viral infections. So anyway, this is just a sampling of the kind of information you can get out of this, and I'll just summarize this. So there are a lot of things that we can do by being able to interrogate the immune system. We can look at autoimmunity, and we found biomarkers for 2 different autoimmune diseases this way. And we're really improving this technology. We can look at viral infection to see if disease causation by particular viruses, sort of epidemiologically. We can distinguish between closely related viruses like Zika and dengue, because we have a lot of epitopes to look at. And we're now looking at the structure of the immune system to ask what different classes of immunoglobulins are recognizing. Are they seeing the same epitopes? And we'd like to do the same thing for T cells. So I'll just stop now and thank the people who did this. 2 postdocs in my lab, Ite Koren and Richard Timms, did the present stability work, and that was based on some earlier work by a postdoc in my lab, Sherry Yen, who now has her own lab. Chi-Kai Shu did the work on transcription with a technician, Yumei Lin, and VirScan was developed by George Shu and Thomas Kula, 2 graduate students in my lab, and a 3rd graduate student, Ben Warman, initiated some of the early technology in that. And then my super technician named Lee, she does everything, so I have to thank her for everything. So I'll just stop and thank you very much for your attention.
At the end of the C-terminal background story, you have cross correlation in there. The regulation of stability, where you have one thing to on and off, not just be unstable all the time or stable all the time. You'll be using these phosphodiagrams or other kinds of modification. Is the C-terminal regulated that way, or?
Well, I think, yeah, so the question is whether or not the modifications, A, would even be detected in this system, and B, are they going to be subject to regulation? And so I think the answer is yes, they'll be subject to regulation there, just like anything else. And but the system as a peptide may or may not get the phosphorylation that would reveal that. But you can often make mutations to mimic phosphorylation, S to E or S to D and T to E mutations. And so we're making a library of the serines at the end swapped out for these phospho-mimics to see if we can't create some degrons that way to go after the phosphodegrons. But the other thing is that, well, okay, so why would you make something that's just always unstable or always stable? You know, what's the point? And you know, I think that a lot of it has to do with whether things are in complexes, and that that if you're in a complex, you're protected, and if you're free, you're not protected, and that's a stoichiometry issue. So that a lot of the ubiquitin ligases are going to be involved in quality control, especially the ones that aren't regulated by some signal. Of course, you can also regulate it by modifying a protein and changing whether it folds out and it's accessible or not.
You started off with the notion that this is a mechanism to quantitate protein within the cell, not the protein. And you showed how degradation and stability. How does one go from, say, lifting? Is it phosphorylation? Is it change? How does that happen?
So how are things are regulated? And I think part of it, as I was saying, was that you want to make sure that things are in complexes and not, if you make too much of one particular protein, you don't get rid of it. It's just going to clog things up. And then we've shown that in our earlier work, I didn't go over this, for the colon ring ligases, in particular, two families of the F-box proteins were WD40 repeat proteins and leucine-rich repeat proteins. If they fold into domains that bind phosphopeptides, so protein kinases come in and they tell the substrate, you know, they change the substrate, then it goes into this general machinery. There are also ubiquitin ligases, like one of the F-box proteins called SCIP2. It cycles during the cell cycle, and so it destroys things when it gets made in S phase, and then those are stabilized. So there are all kinds of different ways you can integrate regulated stability into the system.
There, as part of my question, we are going to put a lot of natural protein into your system, independent cellular system. Can you explain or comment on how the membrane protein, the traffic membrane entry,
so the membrane proteins, what about the membrane proteins? That's the question, okay. And the configuration that I showed you here, where you fuse to the N-terminus of the orth, or the protein, not the peptides, but now you're using the full-length proteins, that wrecks the membrane proteins. So they're almost all unstable, and they get degraded. But we have a system where you can flip it and put the GFP at the end. And then, assuming that it's functional, you can still follow the stability of the fusion protein. But a lot, a caveat with this, when you do the whole protein, is that you can interfere with the function of the protein, and it won't always tell you the truth about the real stability, but it will show you when things are manipulated. So you can take one of these ORF libraries, fuse there, or a peptide library, you can put it into a system, differentiate it, and look for new markers that say, hey, it's changes now that you differentiated into neurons, you differentiated into muscle, and you can use that as a new marker. So there's a lot of uses for the system.
Last question?
Thanks, Steve. Are there any heritable disorders that can be traced to these motifs in the muscles you're describing?
We have not mapped enough of these motifs. I mean, this is really ongoing work. I know that there are, I don't know about heritable disorders. I wouldn't be surprised at all. I know that there are mutations in some of the degron motifs in cancer and beta-catenin and things like that. And so I would not be at all surprised that that would be a source, but they are very small targets. So you might look more for mutations, heritable mutations in the ubiquitin ligases themselves than some bigger targets that would be more likely to be a frequent event.